Hi, my name is Eduardo. Our group project is Comparative Genomics and Evolutionary Insight into Antimicrobial Resistance. Our team is me, Eduardo, Kian, Campbell, and Chang. To detail the abstract, our research team has been investigating the effects of thyroidazine TDZ and dicloxacillin DCX on the genetic mechanisms of MRSA, particularly the US 300 strain. By using advanced genomic tools like GO2R, we analyzed how these drugs impact gene expression in MRSA, individually and in combination. Our results were illuminating. Through gene ontology and KEGG pathway analysis, we identified significant changes in three key pathways related to L-lysine biosynthesis and the transport of proline and betanine, crucial for MRSA survival and adaptability. To provide some context for our study, we are facing a critical health crisis, antimicrobial resistance, or AMR. The World Health Organization identifies AMR as one of the top 10 global health threats. Each year, AMR causes around 700,000 deaths worldwide, a number that could escalate to 10 million annually by 2050 if no effective interventions are made. AMR results from an evolution of bacteria through rapid mutation and horizontal gene transfer, which could alter drug targets and increase drug degradation. To combat this, we use tools like comparative genomics and GO2R to decipher these genetic mechanisms. Successfully addressing AMR requires actions beyond healthcare settings, including addressing ecological and socioeconomical factors to curb the spread of resistance.
On or with DCX can change its gene behavior in MRSA USA 300. It's a type of bacteria that resists to antibiotics. We want to see if this treatment makes the bacteria less resistant to antibiotics. So we will treat MRSA with TDZ and then use a tool called GEO2R to see how the genes in treated bacteria differ from those in untreated ones. We aim to find out which genes are affected by the treatment. After identifying those genes, we will use gene ontology (GO) and KEGG analysis to understand what those genetic changes could mean for fighting antibiotic resistance.
Hi, my name is Campbell, and I'll be discussing the results section today. So to start, it's important to note that there were three important pathways we obtained from KEGG, and the first one is going to be the L-lysine biosynthesis 2 pathway, which is vital for making cell wall proteins, meaning that if this pathway is disrupted, the cell wall could weaken. This would make MRSA much more susceptible to antibiotics that target cell wall synthesis. The next pathway is the transporter in the plasma membrane, which controls the flow of substances in and out of the cell. When this is altered, it could stop MRSA from transporting antibiotics and increasing their cellular concentration and function. And lastly, the protein betaine transporters help MRSA function in an unideal environment, and disruption of this could result in cellular stress, making it vulnerable, and especially when under attack of antibiotics. Understanding these genes and pathways reveal weaknesses in MRSA's defense mechanisms, meaning we should target these pathways. This would result in cell wall weakening, altering its nutrient intake, and reducing its ability to maintain a homeostatic environment. This overall would make MRSA much more vulnerable to antibiotics, which helps in the fight against AMR. This study shines light on the strong potential that TDZ has as a non-antibiotic agent and its ability to combat AMR through disrupting important pathways needed for MRSA's adaptive response. Combining differential expression analyses with functional pathway elucidation forms a basis for further investigation into the therapeutic index of TDZ in addressing the global challenge of AMR.
Hi, I'm Kian, and I'll be talking about our discussion section. So, based on our results, we discovered that thyroidazine, TDZ, affects gene expression in the MRSA substantially, but the important pathways disrupted are the L-lysine biosynthesis 2 pathway and the pathways for the A and the proline betaine transporters in the plasma membrane. These pathways are important for antibiotic resistance in the MRSA, and disrupting them makes MRSA more susceptible to the treatment. Interestingly, the influence of dicloxacillin on gene expression was not as expected, suggesting a unique mode of communication between TDZ and MRSA. Our findings open new avenues for leveraging non-antibiotic drugs like TDZ to combat antibiotic resistance. However, this is a laboratory study, and it's important to recognize the limitations, as it is unable to replicate the body's complex environment. TDZ does show promise in treating AMR, but further research is necessary to determine its safety and efficacy in clinical settings. We believe that in the future it would be valuable to consider developing more comprehensive research addressing the potential of TDZ in overcoming AMR. In vivo modeling techniques and pharmacokinetic studies are essential to validate our in vitro findings. We must adopt a holistic approach to combat the growing threat of AMR, with repeated use of drugs like TDZ offering a potential solution for persistent infections. Thank you.